Body fluid circulation, NCRT based single sentence, part 2. Contents of blood which are considered as cell fragments, platelets. Dash can release a variety of substances, most of which are involved in the coagulation or clotting of blood. Talking about platelets. Blood cells divide of nucleus are erythrocytes. Blood cell divide of nucleus are erythrocytes. The two major types of lymphocytes are T lymphocytes and B lymphocytes. Special cells of bone marrow which produce platelet in human megakaryocytes. Megakaryocyte. Shape of RBC in most mammal is biconcave. Generally, a gene has two alternative forms or version. They are called alleles. According to Mendel, it is called allele. Location of alleles on chromosomes. Same locus in a pair of homologous chromosomes. Same locus in a pair of homologous chromosomes. Number of genotypes can be formed if a gene have two alleles. 3. When more than two alleles exist in a population of a specific organism, the phenomenon is called multiple alleles. Why multiple alleles cannot be observed in the genotype of a diploid individual? In a diploid individual, each diploid organism contain only two loci on the homologous chromosomes. In the homologous chromosomes, each diploid organism contain only two loci. Formula to know the number of kinds of genotypes that can occur for multiple alleles. N into n plus 1 by 2. The ABO blood group system was proposed by Carl Landsteiner. ABO blood group system was proposed by Carl Landsteiner. If n into n plus 1 by 2, in n into n plus 1 by 2, n represents number of alleles. With n into n plus 1 by 2, we can find out the number of genotypes. Chromosome of human which contain loci for Rh factor. Chromosome 1. The chromosomes of human which contain loci for blood group. Chromosome 9. They are actually carbohydrate groups or sugar polymers that are bound to lipid molecules. Surface antigens. Chemically that induce immune response with respect to ABO blood grouping are called. We call them as surface antigens. Surface antigens, nothing but A and B. Surface antigens A and B. These are the surface antigens. We call them as A and B sugar polymers. Antigen present on the surface of RBC of a person with blood type A antigen. Antigen present on the surface of RBC of person with B blood type, B antigen. Antigen present on the surface of RBC of a person with blood type B, AB. In case A and B antigen, both are present. Antigens on the surface of RBC of a person with blood type O, no antigen, no sugar polymers. Person with blood group whose plasma is with anti-B antibodies. Person with blood group whose plasma is with antibodies. A blood group is with B antibodies, A blood group. Person's blood group whose plasma contain anti-A antibodies. B blood group. If anti-A and anti-B antibodies are present in the plasma, predict the blood group of a person, O blood group. If the plasma of a person is without anti or anti-B antibody, such person blood group is AB blood group. Gene named for ABABO blood group. Gene named for ABAB blood group is isoagglutinogen. We call it as isoagglutinogen. The blood group gene I means what we talk about. Isoagglutinogen. Gene name is I. Blood group classification based on the presence of antigens or sugar polymers. We have two types of sugar polymers. They are A and B. A and B sugar polymer are antigen. Possible genotypes for A blood group are IA and possible genotypes for A blood group are AA or homozygous A, heterozygous A. Possible genotypes for B blood group are BB or BO. Possible genotypes for the AB blood group are AB. Possible genotypes for the O blood group, O, homozygous O. Dominant allele among the IA and IO. Dominant allele among the IA and IO. IA. Dominant allele among the IB and IO. IB. Dominant allele among the IA and IB. Both are dominant. Both are dominant. We call it as codominant factor. Codominant alleles of blood groups. Codominant alleles of blood groups A and B. If the two alleles of a character each other, only one of the allele come for expression from the above theory proposed by Mendel. Expressed allele is called dominant allele. The allele which is not expressed is called recessive allele. Expressed allele with respect to blood group A and B. Allele which is not expressed in front of the dominant one with respect to blood groups O. The above theory is named as law of dominance. If the two allelic forms of a character are crossed each other, both try to express and both are succeeded to express. From the statement, this theory is named as codominance. Example for theory, blood groups, particularly A and B. Do it follow Mendelism? No. Number of possible genotypes of blood groups in human being, 6. In type A blood group person, number of genotypes, what are the genotypes possible? Homozygous A and heterozygous A. Antigen present on the red blood cells. Antigen present on the red blood cells, A. Antibodies present in the blood plasma, anti-B. Blood type that can be tolerated. Blood type that can be tolerated, A and universal O. Blood type that can accept the blood. Blood type that can accept the blood. Blood transfusion, the blood type which can accept A and AB. In type of B blood group person, Number one, genotypes. Homozygous B, heterozygous B. Antigen present on red blood cells B. Antibodies present in the blood plasma anti A. Blood type that can be tolerated. Blood type that can be tolerated B and O. Blood types that can accept for blood transfusion. Blood types that can be accept B and AB. In type AB blood group, genotypes AB, antigen present in the red blood cells A and B, antibodies present in the plasma, no antibodies, blood type that can be tolerated, it is universal blood, universal, that means it can tolerate A, B and AB, O. To whom it can give only to the A. In type O blood group, genotypes, homozygous O, antigen present in the red blood cells, no antigens, and antibodies present in the blood plasma, A and anti-A and anti-B. Blood type that can be tolerated, only O. Universal donor it is, it can accept blood type that can accept blood for transfusion, A, B, AB and O. Number of possible genotypes in A blood group person. Number of possible genotypes in A blood group person. 2 and 2 and B. Number of possible genotypes in AB and O blood group person. 1 and 1. Number of possible phenotypes in blood groups. Number of possible phenotypes in blood groups. 4. Number of possible alleles in blood groups. 3. A, B, O. Blood group available in both homozygous and heterozygous forms. A and B. Blood groups available in only single form. A, B and O. The ABO phenotype of any individual is ascertained by mixing a blood sample with an anti-serum containing anti-A or anti-B and anti-D antibodies. This process is called blood group testing. In blood typing, if a clump is formed with anti-A serum only, predict the blood group, A blood group. In blood typing, if clump is formed with anti-B serum only, B blood group. Clump is formed with both anti-A and anti-B antibodies, that is AB blood group. If no clump is produced with either of the antibodies, predict the blood group, that is O blood group. Person with an RH antigen on the surface of RBC are considered as RH positive. Person without RH antigen on the surface of RBC are considered as RH negative. RH negative. Antibodies produced in the laboratory against RH antigen are called anti-D antibodies. Anti-D antibodies. In a laboratory, if clump is formed with a given sample after adding anti-D serum, predict the blood group RH positive. RH positive. If in laboratory, if clump is not formed with a given sample after adding anti-D serum, predict the blood group RH negative. Antigens available in the person who do show agglutination with any serum of a laboratory, that is A, B, then RH positive. Simply we can call it as AB positive. Universal recipient blood group. Universal recipient blood group is AB blood group. Universal donor blood group. Universal donor blood group is O blood group. Qualification to become universal recipient, presence or absence of A, B and RH antigens. Presence. Qualification required to become universal recipient, presence or absence. Absence of A, B and RH antigens. Plasma proteins with AB blood group has no antagonistic antibodies. That does not cause agglutination of RBC received from the persons of other group of blood. So people with AB blood group are called universal donor. RBCs of an O group have no antigen, so agglutination does not occur with any other group of blood. Hence, person with O blood group are called universal recipient. Universal recipient. If a blood group is without antigen A, B, or H, it can become universal donor. If a blood group without any antagonistic antibodies, it can become universal recipient. Antigen similar to 1% in rhesus monkeys, it is also observed on the surface of majority of 80% persons of human is RH antigen. Why the RH factor is named like so? It was identified first. It was identified first in rhesus monkey. Presence of RH factor in the blood, dominant trait or recessive trait? Presence is dominant. Person without RH factor feels RH factor as antigen and show immune response against them, produce antibodies. Why? RH negative persons, RH negative person, immune system feels it as unknown factor. Unknown factor will become antigen. If a RH positive male homozygous married RH negative female, what are the possible phenotype expressed in offsprings? All children are with RH positive. Offsprings of heterozygous positive male with RH negative female, 50% offspring, 50% positive and 50% negative. If a child with homozygous RH positive male and RH negative female is 100% RH positive. Second, third children of homozygous RH positive male, RH negative female, respectively, again 100% positive, RH positive. Generally, the first child of this genetically incompatible marriage is unaffected. During the second pregnancy, when the child is again RH positive, these antibodies enter the fetal blood circulation. The blood cells of RH positive fetus are destroyed, causing HDN, hemolytic disorder of newborn baby. Answer the questions based on the above. Which marriage is considered genetically incompatible? Father is RH positive, and mother is RH negative. In case this is the incompatible marriage. Why first child is unaffected? Why first child is unaffected? Because fetus is delivered by the time delivered the time fetus is delivered, by the time mother gets sensitized. Mother gets sensitized and produce anti-D antibodies. Antibodies produced by the mother are called, as we call now, anti-D antibodies. Type of immunoglobulin mother produced against Rh antigen R, IgG. Why mother is sensitized? Why mother is sensitized? There is no Rh, there is no Rh antigen in mother. So then mother considered fetal Rh antigen as foreign antigen. There is no Rh antigen in mother, then mother considered Rh antigen as foreign antigen. Can we call this as a specific line of defense? If yes, how? Yes, it's a specific line of defense. B cells are involved. B lymphocytes are involved and produce antibodies. They produce antibodies means they'll have memory cells. How antibodies are reached from mother to fetus? Antibodies, IgG, cross placental border, which are very smaller, and enter the fetal blood circulation. The third child will get the same problem? Yes. Second child onwards, hemolytic disorder of newborn baby. What is the solution? How we can prevent second child not to get erythroblastosis fetalis? Then IgG antibodies to be administered. IgG antibodies need to be administered from 28th to 34th week. 28th to 34th week of first pregnancy. This is one of the precaution to be taken over first pregnancy, during first pregnancy. Next, the another precaution can be taken at this time is after delivery. After delivery of first child, after delivery of first child, anti-D antibodies should be administered. Anti-D antibodies should be administered soon after delivery. These two are the precaution methods to be taken over. Anti-D antibodies should be administered soon after delivery. If we give anti-D antibody containing injection to the mother after the delivery of first baby immediately, how it works and how it prevents second child not to get HDN. Okay, so what it will do? This develops passive immunity. This develops passive immunity and prevents the formation of anti-D antibodies. It prevents the formation of anti-D antibodies in the mother's blood, destroying the Rh positive fetal erythrocytes before the mother immune system sensitized. So how it prevents the second child that is developed passive immunity and prevents the formation of anti-D antibodies in the mother's blood, destroying the Rh positive fetal erythrocyte before the mother immune system is sensitized. If she wants third child, do we need to give the anti-D injection again? Yes, immediately after second child. If we give anti-D antibody injection, if we give anti-D antibody injection to mother, anti-D antibodies kill the fetal RBC present in the mother and she cannot be sensitized, followed by she cannot develop memory cells. Is it true? Yes, it's true. Possible time and possibility of fetal blood cells with RH factor. RH factor which may pass and combine with RH negative maternal blood. The fetal blood cells may pass through ruptured placenta, may pass to the ruptured placenta at birth into the RH negative maternal blood. RH negative maternal blood. The fetal blood cells may pass through ruptured placenta. Fetal blood cells may pass through ruptured placenta at birth into the RH negative maternal blood. What is response of RH negative mother when her blood is exposed to RH antigens of fetal RBC? The mother's immune system will recognizes, the mother immune system recognizes the RH antigens, consider it as antigens, RH antigen, and gets sensitized. That means she responds and produce RH antibodies. The mother's immune system recognizes the RH antigen and gets sensitized and produce RH antibodies. Type of antibodies produced by mother against fetal RH antigens, immunoglobulin R, IgG. Most of immunoglobulins cannot pass from mother circulation to fetal circulation. Then how is it possible RH antibodies of sensitized mother can pass through the placental barrier and fetal blood circulation? RH antibodies are IgG type, which are very smaller in size. IgG type, which are smaller in size, which can cross the placenta. If father is heterozygous Rh positive and mother is negative, what is the possibility of first child to get sensitization against the fetal Rh antigen? 50%. What is the possibility of second child to get erythroblastosis fetalis? 25%. Is it possible if they have two children and nothing happened to them? If they are not twins? Yes. Both times, child may get RH negative. Possibility is there. First RH negative or first time RH negative and second time RH positive. This possibility is also there. If you have any doubts, please visit the site body fluid circulation, search as Neet Srikanth. They will find all these problems. 160. Fetus or newborn who are affected with HDFN show hemolytic anemia and jaundice. To compensate hemolysis of more number of RBC, not only from the bone marrow, but also from the spleen and liver. So many large and immature cells in erythroblast stage are released into circulation. Because of this, disease is called erythroblastosis fetalis, hemolytic disorder of newborn baby. In this case, called erythroblastosis fetalis. The father is Rh negative, mother is Rh positive. What is the possibility of HDFN for first, second, third child respectively? First child 0%, second child 0%, third child 0%. These are the possibilities. If father is Rh negative, mother is heterozygous Rh positive, what is the possibility of first, second, third child respectively? Same, 0%. For all the children, 0%. If mother is positive, baby is negative, will HDFN affect the child? No. Here it is. No. If mother is heterozygous positive, baby is Rh negative, will HDFN affect the child? No. Mother is not sensitized. If mother is having Rh factor, there is no problem. Then, if father is and mother are both are negative, so no problem at all. Do not have any other chain. It's affected with HDFN? No. Rh negative mother who is taken blood from Rh positive person earlier and married homozygous Rh positive male. What is the possibility of our first and second child to get HDFN respectively? 100%. Second child, 100%. Both conditions, 100%. Symptoms of neonatal babies who are affected with HDN. Symptoms: hemolytic anemia, they get, and jaundice. Destruction of RBC. Destruction of RBC promotes more production by bone marrow, spleen, liver of newborn, but due to rapid production, many immature erythroblasts are produced, hence called erythroblastosis fetalis. In this case, the disease is called erythroblastosis fetalis. Okay, so then we'll do the blood group inheritance problems in the part 3. Thank you.